Hello again. We've looked at an overview of RNA synthesis or transcription. We saw the role of RNA polymerase and how post-transcriptional processing readies an RNA molecule for its final function, events that are especially significant in eukaryotic cells. In this module, we'll focus on the enzymatic details of transcription in all cells and of mRNA processing in eukaryotes. Processing of other RNAs will be for another module. First, let's look at RNA synthesis in prokaryotes closely. We've seen that genes encoding several proteins of related function are often organized into DNA operons in bacterial cells. Operons are transcribed as a single unit, generating an RNA transcript with several protein coding regions in a single messenger RNA. In bacteria, there's a single RNA polymerase enzyme to catalyze the synthesis of all kinds of RNA made in the cell. Promoters, to remind you, are DNA sequences that mark the start of a gene by attracting and binding the RNA polymerase, as well as other proteins required to start transcription. The structure and sequence of promoters has been worked out in bacteria thanks to old-fashioned and genetic cloning techniques. On the double helix you see in blue here, imagine a protein coding gene to the right of this slide. The double helix is seen as having unwound at the promoter shown in red. The promoter consists of a pair of short DNA sequences called the -35 sequence and the pre-mRNA box, named after the fellow who characterized its function. To be a promoter, these two sequences must be separated by a precise number of nucleotides. But the exact sequence of DNA between the pre-mRNA box and the -35 element is not important. The sigma factor, illustrated as a red oval attached to RNA polymerase here, is required for the polymerase to recognize and bind to the promoter. Once transcription starts, and after the polymerase has moved a short distance along the template, the sigma factor dissociates from the RNA polymerase. RNA polymerase will then continue to elongate a transcript without the help of sigma factor. We'll be looking in some detail at how cells regulate which genes are turned on and off, and how they turn them on and off. Here it's interesting to note one of these ways. Bacteria have evolved different promoter sequences, some of which are shown here, along with different sigma factors, so that genes can be selectively activated or silenced depending on which sigma factors are available. Here we take a closer look at transcription elongation. The RNA polymerase has moved away from the promoter, and a length of RNA in green has been synthesized. The RNA polymerase, very much on its own now, continues to elongate the transcript. As the polymerase progresses, the DNA strands reform a double helix behind the unwound region of DNA being actively transcribed. The promoter is hinted here in red off to the left where transcription began. Termination of transcription is in response to a signal in the DNA that marks the end of a gene. In this example, the signal is itself transcribed, but we know that the transcript forms this so-called hairpin loop near its 3' end. The hairpin loop destabilizes the association of the transcript with the polymerase and the DNA strand, and they come apart. Eukaryotic cells use different RNA polymerases to make different RNAs, and then process their transcripts in several ways. This slide summarizes the complex steps of transcription initiation at eukaryotic promoters. Most eukaryotic promoters include something called a TATA box, with a sequence TATAAA, very similar to the pre-mRNA box in E. coli. We say the promoter is upstream of the transcription start site, by convention to the left of the transcription start site. If you were quick, you saw that the first protein to bind the promoter in this animation is called transcription factor TF2D. This starts a sequence of protein bindings, bringing other transcription initiation factors to the promoter in a specific order. One of the last proteins to add is RNA polymerase II, the polymerase responsible for messenger RNA transcription in eukaryotes. The last step in initiation is a phosphorylation event. The carboxyl end of RNA polymerase II gets those phosphates and becomes active. The activated polymerase can begin new RNA strand transcription. Eukaryotic mRNAs are elongated until Pol2, RNA polymerase II, reaches a termination signal. The characteristics of termination signals in eukaryotes are not as well understood as they are for E. coli. We'll look next at post-transcriptional processing of eukaryotic mRNAs. The discovery of split genes and RNA splicing were quite a surprise, since all clues from studying bacteria had indicated that genes for proteins were just a string of code words or codons that corresponded exactly to the string of amino acids that the gene encoded. The first clue that something might be amiss came from studies of cloned globin genes. Reticulocytes are the precursors to the red blood cells of mammals. They make mostly hemoglobin. At some point, they lose their nuclei and stop making hemoglobin, becoming the familiar flattened red blood cell. When reticulocyte RNA extracts were mixed with cloned globin genes that had been heated to denature the DNA, the RNA was able to hydrogen bond to the DNA, forming RNA-DNA hybrids because of complementary base pairing. By the time of this experiment, it was possible to visualize nucleic acids in the electron microscope and to distinguish double and single-stranded lengths of nucleic acid. Here's how the technique, called R-looping, revealed the existence of so-called split genes. Cloned globin gene DNA was denatured. The extracted globin mRNA, shown in red here, was added. When the temperature was lowered again, some of the RNA strands competed with DNA to form H-bonds with complementary strands of DNA. This drawing illustrates the expectation that the globin RNA molecules should bind complementary or antisense strands of DNA in the solution of cloned globin genes and thereby displace the sense strand. The sequence of the sense strand is essentially the same sequence as in the mRNA, except for thymines instead of uracils. RNA-DNA hybrids did form, but not quite as expected. Here's an electron micrograph of the RNA-DNA hybrid structures that actually did form. The thinner or lighter tracks here are interpreted as single-stranded nucleic acid, and the darker tracks then must be double-stranded nucleic acid. But the double-stranded loop, number 3 here, seems out of place. Here's the explanation. The lighter tracks are indeed single-stranded DNA that were displaced when the mRNA that was added to the cloned gene DNA formed H-bonds with its complementary strands. The dark double-stranded loop, the unexpected bit here, seems to be in the middle of the coding region of the DNA, the part that can form an RNA-DNA hybrid. This big dark loop of double-stranded DNA was interpreted to be non-coding DNA, DNA neither of whose strands was complementary to any part of the mRNA. So it was DNA that interrupted the coding sequence, thereby splitting the gene. So we can conclude that the region of the gene that becomes mRNA must be split into coding and non-coding regions of DNA, and that the non-coding region is not present in a mature messenger RNA. Most eukaryotic genes are split, are composed of what we now call introns and exons. Here's how split genes are transcribed into pre-messenger RNAs and how a pre-messenger RNA is processed to make a mature mRNA with a single undivided coding region. After transcription of the pre-message, complete with all the introns and exons, the introns are removed by splicing, a process analogous to how film directors cut out unwanted parts of a movie and splice together the scenes that will be in the final production. After cloning many split genes and studying their intron-exon structure, it turns out that there's not much to the structure of an intron to explain how it's removed from the pre-mRNA. The 5' end of most introns begins with the bases GU, a GU dimer, called the 5' splice site, because during splicing, the G is hydrolyzed from the end of the exon at the left. The 3' end of the intron is an AG dimer. Again, during splicing, hydrolysis separates the intron from the second exon after the G. One of the many adenine nucleotides within the intron, one usually closer to the 3' end than the 5' end of the intron, is important as a branch site that is used in splicing, although it's not clear why any particular A serves this function. Splicing itself is catalyzed by a group of small ribonucleoproteins, small particles made up of RNA and protein found in the nucleus, called SNRPs for short, or snRNPs. Here's how they work. First, snRNP U1 and U2 bind to the 5' splice site and to the adenine at the branch site, respectively. Then U4 and U6 associate with U2 at the branch site. After U5 snRNP binds at the branch, the 5' and 3' splice sites are brought close together in a structure called the spliceosome. The next step is cleavage of the 5' splice junction and the formation of a bond between the 5' G of the intron with the branch site adenine nucleotide to form a lariat-like structure shown here. This is followed by the hydrolysis of the 3' splice site and the release of the lariat, concurrent with the ligation or splicing of the exons. Upon completion of splicing, the spliceosome disassembles and the snRNPs can be reused to splice again. So, what are introns good for? When did they arise in evolution? Where did they come from? Were they part of genes from the get-go in the first cell in the progenote? Here's what we know and what biologists are arguing about. Introns early. This is an hypothesis that says that genes in the progenote arose complete with introns, but that when eukaryotes and prokaryotes split in evolution, the prokaryotic organisms lost their introns, that is, their genes lost the introns, while the introns were retained in the eukaryotic cell lineage. Introns late. This hypothesis says that since most eukaryotic genes and some archaebacterial genes are split, introns were acquired after the divergence of these domains from bacteria, perhaps from viral infections or a transposon invasion of genomes that inserted introns into genes. This might shed some light on the argument. The genes in some organelles, mitochondria and chloroplasts, have introns. Even more surprising was the finding that these introns in many cases were self-splicing. That is, they did not use a spliceosome-like mechanism or other enzymes to bring the exons together and to ligate them. Could these split organelle genes be left over from the endosymbiotic events that created mitochondria and chloroplasts out of bacteria and cyanobacteria? That might imply that bacteria still had intron-containing genes at the time of endosymbiosis. If this were the case, would this support the intron's early notion? Well, the debate is not settled. These self-splicing mRNAs were the first RNAs to be tagged as ribozymes, that is, RNAs with enzymatic activity. We'll see at least one other example of an RNA with an enzymatic activity shortly. The first cells may have combined catalytic and information functions, not to mention energy storage functions, in the same nucleotide-based molecules, and in particular in RNA. So the first genomes may have been RNA and not DNA. This slide suggests that self-replicating RNA genomes in the earliest cells evolved a catalytic function enabling polypeptide synthesis. Then later, DNA genomes evolve, perhaps because it was beneficial to organisms to separate the protein-making functions of a cell from the informational or genetic functions of replication. Clearly, introns are not necessary to the function of a messenger RNA. Bacterial genes don't have any. What's more, intronic DNA is often much longer than the exons of a gene, implying perhaps a lot of wasteful DNA that has no apparent function. So, why do introns exist at all in eukaryotic cells? Or why, if they were present in the first cells, were they retained by eukaryotes and lost in prokaryotes? Well, let's recall protein domains and protein families. The existence of introns permits easy recombination of exons, resulting in a movement or a shuffling of exons in and out of genes that didn't have these sequences before. By this mechanism, proteins could rapidly acquire new domains and thereby new functions. Today, we can see a relationship between many exons and the structural and functional domains that they encode in polypeptides. Here is a generic example of exon recombination or exon shuffling initiated by unequal crossing over between two different genes. First, we need to understand that crossing over, both equal and unequal, are common events in eggs and sperm, or during the development of eggs and sperm. Most of the time, we do not see the damaging consequences since the organisms don't survive. But recombination within introns increases the probability of survival of unequal recombinant organisms. After unequal crossing over, exon C in this example is now part of a gene that previously only had two exons, A and B. This is now a 3-exon gene. A 3-exon mRNA is made by splicing, and a protein then with a new domain will be translated. The recombination event could have occurred almost anywhere within the non-coding, that is, the intron DNA. This imprecision makes the occurrence of these recombinations more likely than not, allowing rapid gene and protein evolution. In addition to splicing, eukaryotic mRNAs are further processed before they become mature mRNAs ready to be translated into polypeptides. They get a 5' cap and a 3' poly A tail. The 5' cap is a methylated guanosine nucleotide. This cap helps move mRNA out of the nucleus and into the cytoplasm. Near the 5' end of the mRNA is a sequence that recognizes an enzyme called poly A polymerase. That was the blue molecule shown here, which then binds and catalyzes the addition of several hundred adenosine nucleotides to the 3' end of the transcript. This poly A tail seems to govern how many times this message will be used to translate a polypeptide. In vitro translation experiments show that the poly A tail gets shorter and shorter the longer a solution of mRNAs is allowed to translate proteins. Here are some capping details. A capping enzyme brings a methylated guanosine nucleotide and an mRNA molecule head-to-head, as shown here, the check marks being the nucleotides. The enzyme then catalyzes a condensation reaction to form this phosphotriester linkage. In this illustration, the cap and poly A tail recruit binding proteins from the nucleus. One, shown here as a brown crescent, is a cap binding protein that binds to the 5' end of the messenger RNA. The cap protein, together with an RNA export factor, shown as the red square here, guide the messenger RNA out of the nucleus through a nuclear pore complex. Once the mRNA is out, the export factor comes off and can return to the nucleus, while a translation initiation factor displaces the cap binding protein. The cap binding protein probably goes back into the nucleus to be recycled as well, although that's not shown here. A poly A binding protein, the green oval in this slide, binds to the 3' end of the message while it's in the nucleus, and then remains bound in the cytoplasm. And that brings us to the end of this module.